Hello, listeners. Yamina here. I'm very excited to have you join us for another Dr. GPCR podcast episode. If you haven't heard, we are officially a nonprofit 501c3 organization. Becoming a nonprofit only solidifies our mission to serve the GPCR community. Therefore, we're taking a short podcast break starting in July to come back with new episodes at the end of August, as well as an amazing set of events in the fall. If you are an ecosystem member, by now you know that every Thursday you get your weekly news or weekly GPCR news delivered directly in your inbox. If you're not an ecosystem member yet, join us today. Thanks to our fantastic team of volunteers of GPCR scientists, a GPCR article will also hit your inbox weekly on Tuesdays. Also, mark your calendars for not one, but two Dr. GPCR University classes with Dr. Terry Kanakin. The first one is titled Techniques for Effective Lead Optimization of Candidate Molecules, Principles of Pharmacology for Drug Discovery, which will start on Thursdays, September 5th at 10 a.m. EST and run for 4 consecutive weeks for 90 minutes. Then, on October 3rd, an advanced class will start about therapeutic translation of GPCR activity, advanced method for the optimization of candidate selection. This class will run for 5 consecutive weeks, also from 10 a.m. Eastern Time on Thursdays. For both courses, we will be offering 5 scholarships per course to GPCR scientists who live and work in developing countries. So stay tuned. Just a short week after the beginning of our second Dr. GPCR University class in the fall with Dr. Kanakin, on October 11th, we will be hosting our Dr. GPCR Symposium on GPCRs as therapeutic targets. During that day, we'll have talks from speakers of all levels, both from industry and academia, a poster session, as well as a panel discussion. We're also excited to be co-organizing our very first in-person Adhesion GPCR workshop in collaboration with Dr. Anthony Boucard from the Adhesion GPCR Consortium. Mark your calendars for October 23 to 25 and join us in Mexico City for a fantastic meeting on all fascinating aspects of adhesion GPCRs and more. If you'd like to sponsor the Adhesion GPCR workshop, please email us at hello@drgpcr.com. Both emails, actually, we have now 2 emails, hello@drgpcr.com and hello@drgpcr.org. Both of these emails will be active for a foreseeable future to make sure that we don't miss your emails. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. I'm very excited today to have Dr. Brian Arey. He is a director and leads discovery and optimization at Bristol Myers Squibb in Princeton, New Jersey. Hi, Brian.
Hi, Yamina. Really great to be here. Thank you.
Thank you so much for coming on board. I know we've been planning this for quite some time, and I've dropped the ball multiple times, but I'm glad that we're doing this on this beautiful, fall day. So how are you doing?
I'm great. Thank you. Again, it's the day before Thanksgiving, so sort of winding down today. I'm looking forward to a little break. It's been a really busy and crazy year, as you all know. So I'm really looking forward to a couple of days of relaxation here.
I am too. I'm excited that we're doing this just before Thanksgiving. I'll have the time to, really enjoy the weekend and make sure that, we capture as many GPCR stories as possible before the holiday. So tell us a little bit about yourself.
Okay. So my name is Brian Arey. I'm Director of Mechanistic Pharmacology within the Leads Discovery and Optimization organization in discovery research at Bristol Myers Squibb. I also am interim director currently of the compound management and core automation teams. So overall, those 3 organizations comprise about 70 people right now. So it's a fairly large remit, but I really enjoy it. I've been in drug discovery research for almost 25 years now. I started in actually 26. Starting back in 1994 with Wyeth. And I've worked in many different aspects of drug discovery, everything from in vivo pharmacology model development to molecular biology, and many different therapeutic areas. So I really have a sort of a broad sort of view of drug discovery across many different physiological systems, which has always sort of been my thing, so to speak, in terms of really providing value to the organization.
That's great. Actually, you answered my next question. How many people work for you?
Oh,
70
yeah,
people.
yeah.
How do
So
you juggle that?
it's not easy. So, and well, truth be told, the management of the core automation and compound management teams is on an interim basis. I've been doing it for about 3 months now, and probably will be doing it for another 3 months. But I've really, really enjoyed it. Checked the box on my career bucket list. I've always wanted to lead a large team, and it's been really cool. It's required a lot of learning, and so it does take away from your science a little bit, because there's a lot of personal things and fires to put out on a daily basis. But again, I think it's really about embracing opportunity to do different things, and that's always really sort of spoke to me, and things that I've enjoyed doing, things that I wouldn't have normally set out to do, but have sort of taken on and really enjoyed doing them.
That's great, because so you've had a different experience that allowed you to take on additional responsibilities, but at the same time, you're in a job where you get to take on the reins on something new, and you're given that confidence, but also that appreciation when people tell you, "Hey, Brian, I know you've never done this, but I know you can do
Correct.
it."
No, you're totally right. And it's really, that probably is the most satisfying, right? Because we all want to be appreciated and have impact. I don't care if you're the janitor or if you're, the CEO. That's a sort of a, human nature kind of thing, I think. And
Yeah.
so, and that's what I try to actually instill within the people that report to me. Personal growth and career development are passions of mine, to really help the younger generation learn and grow and develop their careers. It's a lot of fun.
So you mentioned that you've been in industry for the past 26 years. Where were you before? Can you take us back to
Sure.
15-year-old Brian, who may or may not loved science?
I always love science. So that's, I really enjoy science. So, when if you wanted me to go way back, I can go to prepubescent Brian, but but I don't know if you want to go that far. So in high school, I grew up in a small town in Southern Illinois, outside of St. Louis. And I've always had an interest in medicine and physiology, even from an early stage. My father and I, and science too. So my father and I used to watch these television shows, documentaries, all the time, and that sort of piqued my interest. But I also always just sort of gravitated to that level. I mean, when you're young and you're in elementary school, you have very sort of rote specific topics like math, right?
Yeah.
Science and that kind of thing, English, but science was always something I really, really enjoyed and had some sort of, talent at. So I did really well there. And then when I got into high school, I started to pursue that a little bit more with the eye of perhaps becoming a physician. And so when I went to college, I was, that was my goal. And I did okay. I was never like a stellar student, to be perfectly honest with you. I was a B student. I never really, knocked it out of the park, but I didn't really care either. I was always more interested in learning as opposed to getting an A on a certain, exam or something. But what I found was the way I got into science was actually kind of interesting. So when I was a sophomore in college, I had the opportunity to start washing glassware in a research lab just for some spending money as a work study student. And so this was in Dr. Mary Rue's laboratory at the medical center at St. Louis University, where I went to college. And so I spent about 6 months washing glassware. And then, of course, that never actually went away. I washed glassware for the next 3 years. But anyway, when summer came around, she asked me if I wanted to do some research over the summer. And I was like, yeah, this would be great. So I started doing research. And again, I just had a knack for it. And I really sort of enjoyed it a lot. I wasn't doing anything crazy complex. I was basically purifying estrogen and progesterone receptors from tissues and then looking at binding assays. You know, it wasn't, but it was, to me, it was like, you know, so cool to be on the cutting edge,
Of course.
and doing that kind of stuff, right? And as a
Sounds
19
like when you're that young, sounds like magic. Like you're doing the
it,
most.
yeah, when you
The
get your
world's
first binding assay that works, you're like, wow, this actually works. So, and then from there, you know, I really wasn't sure after I had that experience, and I really enjoyed it, and we did some really cool things there. She was studying actually how estrogen and progesterone receptor bind to chromatin, and the role of other, you know, transcriptional complex proteins. So it was actually kind of cool. But the when it came to graduate, I really did. I realized at that time I didn't really want to be a physician, and I really wanted to be a scientist. And so I spent a year and I worked in a lab at the Children's Hospital at St. Louis University, working for this woman named Kathleen Schwartz, who was a gastroenterologist, and she was working on Reye's syndrome. I don't know if anybody remembers that anymore, but that was a big thing back in the mid 80s. Really, what happening was that children who had this syndrome would develop this syndrome if they had like chickenpox and were given aspirin. And they would go into this syndrome where they would have liver failure and die. It was actually a real problem. So anyway, I was a technician there, and I worked in her lab about 2 or 3 months, and actually defined some really key data that showed the mechanism of how aspirin was actually leading to liver damage through the process of chelating iron and then in creating free radical oxygen species. So she came to me, it's like after 3 months, she came to me and she said, Brian, I think you're wasting your time here. You need to go to grad school. So she pushed me to go to grad school, and then the rest is, so I went to Florida State, which I know is not a stellar, you know, it's not like Harvard, but I went there specifically to work with this guy named Mark Freeman, who was a neuroendocrinologist. And I really, you know, at that time I was really interested in neuroscience and endocrinology. Endocrinology always spoke to me, even when I was in high school. And so this was a way to marry those two interests. And so I went there, and he was actually a fairly big name, you know, within the scientific community of neuroendocrinology at the time, and had a great experience there, where I stumbled upon a circadian rhythm. So I was really a physiologist by training. So I stumbled on a circadian rhythm regulating prolactin secretion that actually is still quoted today, that work.
Wow.
30 years later. I'm really proud of that, because I mean, how many people do you know that still have papers quoted? I mean, I just had a citation just last week, so on that work. So I was really stoked about that. But so that was really the first clearly defined research that showed that circadian that hormone secretion from the pituitary could be regulated by the circadian clock.
That's fantastic. I remember having metabolism and classes on hormones and how these things function, and I just find it so fascinating that, you know, that little hormone that you don't think of just can mess up everything in your system, and it's so hard to get.
You can see that theme throughout my career, right? So even as an undergrad, right, you know, working on estrogen and progesterone receptors, then working on neuroendocrine systems. And then when I went to Wyeth, I went to postdoc at Northwestern, which was a cool experience, where I was working on the signaling pathways of prolactin and growth hormone secretion. And I was in Dan Linzer's lab there in the biochemistry department, and that was a cool experience, with the exception that I was competing against a guy named Paul Kelly from France, who had this huge lab, and I was the only one working on the signaling pathway in Dan's lab. So I was just getting slayed. I was like, I was really frustrated.
True David against Goliath situation.
Well, it just seemed like every time I would come up with an observation, I would find it in a paper the following week. It was just like, it was really hard.
Well, you can look at it different ways. Yes, it's hard, but at the end of the day, that just means that, you know, you were on the right path and you had the right thought process. I have a quick question regarding your work on the aspirin and, you know, its effect in children. So was it because these children were getting too much aspirin, or was it because they were just too young and they just couldn't handle the aspirin at all? It may be a naive question there.
No, it's not a naive question at all. It's actually a really, really good question. the question, and I don't know if they ever quite figured that depth of mechanism out, to whether or not there was something about, this was 1986. So, the molecular biology and understanding of cellular function was quite remedial as compared to today. So, but I will say that really the hypothesis that Kathy had was that somehow the aspirin was chelating iron, and that aspirin iron chelate was somehow stimulating free radical oxygen species, because it was well known that iron can do that within a cell. And so what I did was I came in and actually developed the assays to show that hypothesis. It was really Kathy's hypothesis, not mine, but I was able to actually, she had spent a lot of time trying to figure out how to develop methods to show this. And so I came in and, it was just a matter of looking through the literature, and there were in areas that you wouldn't normally look at, and finding, well, there's this assay exists, let's try that, and applying it. So yeah, but so I don't know if it's necessarily a case where they had too much aspirin as opposed to their liver was just not quite fully developed to handle the free radicals that were developed by these chelates.
Wow. Yeah, it's just having a 5-year-old and always being told, especially when they're young, go with the Tylenol and not with the aspirin.
Well, that's the reason. That's the reason. Yeah.
That's always the thought. And we've never, I don't think we've ever given my son other than Tylenol. And it was so interesting. The first time he had a fever and I gave him that 2.5 mils of Tylenol, he was tiny. And I think 20 minutes later, everything calmed down. So I can only imagine if you give such a young child aspirin and dramatic effects. That was my aspirin question. So now you're going to grad school. You're working on prolactin effects.
Yeah. So in grad school, that circadian rhythm work, we published almost a dozen papers on that. So we did everything from the discovery of the rhythm, and I did that mostly independently, with Mark's direction. But that was a great lab. It wasn't really a big lab, but it was me and Mark, a couple other grad students. Tom Burris, who some people might know here, is a well-known nuclear hormone receptor guy these days, and I were grad students together. As it turns out, Tom grew up about 30 miles from where I grew up. It was kind of interesting. We ended up in the same lab, but but there was so I would have to say that Mark was a huge influence on me on a number of ways. And one was, he was tough to work for. There was no quarter given. He expected excellence at all times. He really did. And I think that helped me truly, because at that time in my career, I was kind of my personality is kind of be really casual, and he really taught me just discipline and attention to detail. So I really give him a lot of credit for that. And these are the little things that you find out you need in order to be a successful scientist, right? But he also taught me this concept that techniques are simply tools in a toolbox to answer scientific questions, and that was another key takeaway. But there was a postdoc in the lab by the name of Bela Kanichka. He was from Hungary. And he and I spent a lot of time, late nights together, because the circadian rhythm, I had to take blood samples all night because we didn't have a reverse cycle room.
Of course.
So I would spend all night, literally from 10 p.m. in the evening to 6 a.m. in the morning, taking blood samples from rats in the dark. So Bela and I spent a lot of time, and he really taught me appreciation for GPCRs as well as pharmacology. And he was my first introduction into pharmacology and taught me a ton. And so I have I just wanted to give him a shout out, because he had a huge influence on my career too, just teaching. He was a great mentor, and he's still a really, really good friend of mine now these days too. So then I went to postdoc, did that for a while. And then I got a call about 2 years into my postdoc from Francisco Lopez. Andres Negra-Villar had just started a new institute at Wyeth and the Women's Health Research Institute. And Paco called me up and said, hey, do you want to come work for me? And Paco and I had crossed paths as a because of my graduate work. So, at that time I had a small child. I was living in Evanston, on NIH scale was $18,000 a year.
I feel the I've been at NIH, so I do feel the
So I was an NIH postdoctoral fellow, even though I was at Northwestern, and so I was, at that time I was a pretty prestigious position. And so, but at the same time, you have to make money and support your family. So this position the opportunity to work with Andres was and do that in a pharmaceutical setting was too compelling to pass up. And I knew at that time that I kind of was, pigeonholing myself to a certain extent, that probably I wouldn't be able to go back to academia, which is still very difficult as an industrial scientist. But at that time, it was even more difficult. And there was a lot of, I'm sorry, but bias against industry scientists.
I think there still is in some ways, not as much as 10 or 15 years ago.
Clearly,
but
it's gotten better.
But sometimes I still hear, "Oh, you're on the dark side."
It is. It's really a bias, and I don't understand it. But at the same time, nowadays we do so much work with academic partners. It's and the lines have been blurred significantly. Because every academic scientist wants to start a biotech company. It seems like.
Exactly. Plus, I think it's just two different worlds that complement each other so well.
Yeah.
Because one has, in academia, you do have that freedom to think about problems that may or may not attract funding. I'm thinking of Brian Kobilka, who, when I heard his Nobel Prize lecture, where he had the columns stored in his office to purify, the receptors, to look at structures. But at the same time, when you go to industry, you do have a different thought process, different priorities. But at the end of the day, you have the funding as well to take, that research into the clinic and into, approving it through the FDA, which takes a lot of time, a lot of money. So I think it's
really just applied science. That's really what it is, right? It's applied science. But I knew that it was a hard decision to make, to be honest. And I wrangled for about 2 weeks trying to figure out what I was going to do. But in the end, it was just, I couldn't pass it up, because I knew that the likelihood of this opportunity to get your foot in the door in industry was not going to come down the pike every day. So I simply took it and ran with it. And that's where I actually got really into GPCR work when I was at Wyeth. And that's how I got into signaling bias. I was working on the follicle stimulating hormone receptor, and I was leading a drug discovery team there for agonists and antagonists for contraception and fertility treatments within the Women's Health Research Institute. And so serendipity in science is really, really interesting. And it's really, I think science rewards those people who have their eyes open and have an open mind. So what happened was we created a high throughput screening assay, just looking at cyclic AMP production in CHO cells. But as a standard, we needed to have a reliable source of FSH. And so we created insect cell expressed FSH. We were purchasing pituitary purified FSH, but it was incredibly expensive. So we created this recombinant material. And it just so happened, we didn't realize it at the time, that insect cells don't glycosylate FSH to the same degree as mammalian cells. So when we put it in our culture, we saw this bell-shaped dose response curve, and we thought, well, this is really interesting. What's going? Because the purified material from pituitaries gave you a sigmoidal curve. So really, there's something going on here. So long story short, we ran a simple experiment where we treated the cells with pertussis toxin, because I had this crazy idea that maybe we were also signaling through GI. And I actually got that idea from Tom Burris's work on the dopamine receptor when we were in grad school. So this is another thing. You take things that sort of and use them as you go along in your career. And lo and behold, it turned a bell-shaped dose response curve into a sigmoidal curve.
That is so awesome. I think every pharmacologist who loves sigmoidal dose response curves is now just smiling like I am. It's just phenomenal. Yeah,
it was like the coolest thing ever. I have to say, I didn't sleep for like 2 days. I was so excited, because then I knew that there was really something up. And then we published that work, and that work is still published today, or cited today too, which is really exciting. So what we did from there was, it was about a 40-year-old sort of conundrum in the glycoprotein hormone field as to why glycoprotein hormones were glycosylated and secreted as different glycosylated isoforms that circulate. And so basically, we proposed, and that was ultimately validated by others, that actually the reason for that physiologically is because they signal differently, and they induce a different effect on the granulosol in the case of FSH, or the fecal cell in the case of LH. to drive physiology. That was really cool.
Yeah, I find the human body just fascinating, and you know, zooming down on GPCRs and bias signaling and having all these you know, variables in the body at the same time, but us understanding that little difference in glycosylation patterns is just phenomenal. I think it's just and I totally understand that you didn't sleep for 2 days. I wouldn't have slept either. It's just
but the problem was a lot of people didn't believe me. So this is 1995. And so when we published that data in 97, I got a lot of pushback. I mean, there was a lot of people who said I was crazy. I didn't know what I was talking about. Literally, one guy told me I didn't know what I was talking about. He subsequently apologized, but you know, I took a lot of grief in the literature, you know, people writing reviews telling me I was crazy. But, you know, if you're going to be a pioneer, you got to sort of expect that response, you know, because you're really refuting dogma. And that's never, it shouldn't be easy, right? I mean, if the scientific field has a certain understanding, it shouldn't be easily overturned. You know, you need to have the breadth and depth of data to really overturn that. So
exactly.
You know, it took a long time to get there, but ultimately people came on board, and now it's sort of you know, people see it that way. So, but again, that's another experience, again, just through sort of just following data and keeping an open eye, where you can really sort of see novel findings. And I think that would be my advice to young students, really, is to always keep an open mind.
That's so interesting, because I feel like when you're a scientist and sometimes you have these hunches of, oh, you know, let's look at what PTX does on a signaling pathway. But at the same time, if you follow the science, you ask the right questions, you have the right controls, and you're sure of yourself, some of the data that you get, and you believe the data, and you've, you know, went full circle, then that's when you make the most important discoveries.
No, that's right. So when I was getting lambasted in the literature, you know, I called my graduate advisor up for advice. I said, because I was just distraught. I'm like, I don't know how to handle this. I was really young in my career, and I, you know, thought I was ruined. And I said to him, I said, you know, ask Mark, you know, what should I do? And he said, well, Brian, he said, if you've done all the controls appropriately, and you've done the experiments to the best of your ability, and you've asked all the hard questions, and you still get the same result, then you have to have confidence in your data. And that has stuck with me my entire life, you know, since then, because he's absolutely right. You have to have confidence in your own work.
And data doesn't lie. Numbers don't lie.
No, they don't.
There's no bias there. I want to take you back a little bit to your first interaction with your Hungarian colleague about GPCRs and pharmacology. Can you tell us a little bit more about what you've learned, and did that interaction, that knowledge, have any effect on your next position at Wyeth?
Oh, so absolutely. So the stuff that I've learned from Bella has followed me in my entire career, and that's why I give him a lot of credit for my success, and he knows that I do, because I've done it in a lot of ways. but you know, because I had no knowledge of pharmacology, and certainly receptor pharmacology at all, but he was really sort of a trained pharmacologist, and so as well as a neuroscientist, and so he was working on the endothelin receptors and understanding how endothelin receptors could modulate pituitary hormone secretion. And so he was doing a lot of pharmacology and looking at different antagonists and agonists and how they affected both in vitro and in vivo hormone secretion from the pituitary gland. And so from watching him and talking to him, I learned a lot. And you can learn a lot from people who are doing different things than you, and apply that to your own work. And I sort of soaked it up with a sponge. But the way he presented it was always so interesting to me. You know, it was really never sort of, he never approached it in an arrogant way. It was always like, you know, yeah, so I'm doing this, and this is why I'm doing it, and this is, you know, and so that was my first introduction into GPCR work, and then watching Tom with the dopamine D2 receptor and the work that he did was also sort of an eye opener. You know, again, that was another serendipitous finding as well for Tom in the lab, and that got him started on his way too. So it was a small lab, but we were powerful. We had a lot of impact, I think.
That's great. That's great. And then the position that you got at Wyeth, did you already know ahead of time that it would involve any GPCR related work?
I did, yeah. So I knew that they wanted to work on FSH, and that intrigued me, again, because I had been working in a reproductive biology lab. I mean, it was neuroendocrinology, but it was really focused on reproductive biology, hormones. And so that was sort of a way to link my past with my future. And so I started working on FSH. But in the end, it was really, I went into it just purely with the interest of developing drugs, and finding that exciting and to help patients. And I'm really focused on patients and helping them. And I thought that was a really cool opportunity to marry science with helping people. And so that's why I took that job in the end. And, lo and behold, I stumbled on this thing. And then that really has got me started in the signaling bias field. And I sort of have followed GPCR work throughout the rest of my career. So I've always sought out and linked my career with GPCR pharmacology from then on. And even now, I work on GPCRs. So I work on a number of them. I can't tell you what they are, but,
yes.
but I work on everything from GPCRs and all across many different therapeutic areas. One of the cool things about my job now as the director of mechanistic pharmacology is that the team is really sort of broken up into three research areas. One is enzymology.
Mm-hmm.
The other is GPCR pharmacology and receptor pharmacology, really, because we've broadened the base beyond GPCRs. And that's another thing that I'm really passionate about. And the other is the new field of protein homeostasis. So we work on protein degradation pathways as well.
That's great. I love that you brought it up, that you're still working on GPCRs, because there is, since, bigger companies or even biotech companies usually are in kind of stealth mode and they don't want to divulge what they're working on. You always have, most people talk about, immuno-oncology or, immunology or all of these fields or neuroscience. But when you're, a postdoc or you're someone in the GPCR field and you'd love to transition to what we called previously the dark side and work at a biotech company or even at a big pharma, it's very hard to figure out, okay, how can I contribute? And
Correct.
Are they working in the GPCR field? Or how can I take the knowledge I have? And you look at job descriptions and it's very rare that you see the keyword GPCR. You may see transmembrane proteins, but you never know that. So
yeah, you know why, Yamina? Because these things in industry are like any other social dynamic in human nature, and that is they ebb and flow with popularity. And so what happens is, this is what I've learned over 26 years, is that what is old is new again. And so what happens is when the new sexy thing comes down the pike, everybody runs to that. I've never done that. I've always run away from the crowd, or tried to anyway, because I feel that's where the most and the best opportunity to have impact and to really discover new things is. And so because what happens is fields become very, very crowded and it's really hard to compete. And I think perhaps my experience from my postdoc has, at least subconsciously has impacted me in that way, because I always do try to do things and bring a different angle. And I would have to say that I've always tried to look at it from a physiological perspective or a pathophysiological perspective, because my background really is in vivo pharmacology and physiology. So even though I work in a very mechanistic world these days, it's in the end, we're still trying to just figure out physiology, right? I mean, whether it's you're looking at a signaling pathway or how a protein is degraded, whatever, again, we're just trying to understand in a reductionist sort of way how physiology happens and how it works.
And how can you get it back on the right track so that you can restore health, or at least modulate that pathophysiological event?
And my personal philosophy about drug discovery is that I think a lot of people are always looking for the hammer. They're always looking for that that one thing that's going to cure a disease. And I think what we found, at least over the years that I've been doing this, is that most diseases are multifactorial in nature, and finding that single bullet that's going to cure a disease is very, very difficult and rare. because human physiology is so disparate across cultures and genders, and and that's just based on evolution, right? I mean, that's the evolutionary process. It's not supposed to be the same. From that perspective, I think looking for modulators, things that can modulate a system where you can combine them, maybe with a hammer, is perhaps a more safe approach. And so that's my personal philosophy. And so one of the things that I try to push forward in my career. So if you need to, impact can be given in many different ways. And even though you may see yourself as a GPCR pharmacologist, that doesn't necessarily mean you can't learn new things and apply what you learned as a GPCR pharmacologist to those new aspects.
Agreed. I think no one can take away from you that ability to analyze the data, to understand new technologies, new techniques, to understand new physiological pathways, and be able to zoom out from that little experiment, that tool that you've been using for 10 years, and have a broader view. And I think that's one of the advantages of having a PhD and going through the lengthy training that you go through, because then you can basically do anything.
That's correct. No, you're totally right. And within that, and I think for young people, they need to understand that there are many different opportunities within a pharmaceutical company, outside of even research, where you can apply your knowledge and your growth. Translational medicine is a huge unmet need in most pharmaceutical companies. We do a lot of it, but we really need to make progress there, in my opinion, to make it better. That's one way. Those kinds of things. There's always opportunities to provide impact and
outside the lab, and even in regulatory affairs, where
correct.
You need to understand, the way the FDA thinks, the way all of these happen. But at the same time, if you understand the science and the mechanistics of these drugs that you're trying to get through into the clinic and into people, you absolutely need and use that scientific background.
Absolutely, without a doubt. And
so
we work with those people daily. So anyway, go ahead. Sorry.
No worries. I wanted to take you back a little bit. So you had mentioned that you had a young child going into industry. And as I mentioned before we started recording, I don't think we have ever tackled this question of, building a highly successful career. You've been in industry for 26 years. You still have papers that are cited that, were published when I was barely 10 years old. But at the same time, so how did you juggle that?
Yeah, so that was there was a period of time when I was a single father with two small children, actually. About 4 or 5 years into my career in industry, and that was a very difficult time. And I do think I don't know if I did it great. That balancing act. But really what you have to understand on a personal level is that your career is important, no doubt about it. Your children are more important, and they should be your focus. And so that was the approach I had. So there was a period of time before I got remarried that where my children were my focus. So from about 1998 until about 2002, I did my work and I did some stuff, but I really tried to make sure that my children knew that they were my focus, because at that point they were really young. They were 3 and 5-ish. So those are really important years for them to understand, and that their parents care about them and really want to promote them and support them and want them to do their best. So I think that I wouldn't redo it. If I had to do it over again, I would do it again that way. Because I have an incredible, my oldest son's now buying a house as of today. Actually, he closes today. He's 30 years old or 29, and that's we have a friendship. It's not we have a really close bond, and my both of my sons and I have a really close friendship now, and that's kind of cool, without a doubt. So I would do it again. But I don't know if I necessarily did it well. I just focused on my kids and did my work as best I could. That was, it's not easy. And I see a lot of young people, who have kids that are building their careers, and that's my advice to them always. At this early stage, you have to have, you have to put your focus on your children.
And it's hard. It's hard to juggle in general. And I think, especially here in the US, being a PhD student, it's very rare that PhD students have children. postdocs maybe, but even that is sometimes, you know, not as much. And I was talking to Alex Hauser from Europe, and he has 2 children. He's a postdoc, and I think the support that you get in Europe as a postdoc to have a family is very different than in the US.
Yes.
It's not impossible to have a family as a postdoc or have being, you know, a young scientist, but it definitely is a juggle. And those years you mentioned, you know, 3 to 5 are so precious because they do. I feel like when from the moment they're born, every week they do something new. And it's the most trivial thing, but being able to open a small box and you're like, yay, you did it.
Yeah, exactly.
Or, you know, they get dressed on their own and it's like the most beautiful thing ever, but we do it every day and we don't even pay attention to it.
You know, so that's another personal, that's, you know, so you have to be true to your personality type, I think, you know, and be really true to yourself and know what your strengths and weaknesses are. You know, for me, I'm kind of a nurturing kind of guy. Even, you know, even with the people that report to me, I take them, I see it that way, that I'm trying to nurture their careers and their career development. And I think that, you know, it was okay for me, right? You know, it was okay for me to maybe take a step back from the science for a little bit to take on that, because that helped enrich my life in a way that I couldn't enrich it through science. When I wrote my book, you know, I dedicated it to my children, and I remember I got a few copies of this book, and I gave each one of a copy, and really what I said to each one of them was that when I scribbled a little thing in there, because I wanted them to remember me when I'm gone, and I said, you know, I wrote this book for you, which was true. I wrote it for them so that they understand what I did and what my science was about, because none of them are scientists. And then I said, you know, thank you for enriching my life, because that's really how I felt. I mean, you know, they add things that you can't get anywhere else.
I couldn't agree more. I mean, I have, as I mentioned before, I have a 5-year-old, and I feel like, although he's only 5, and I cannot imagine my life without him.
Correct.
It's like my life, our life before him never existed, because it just brings, yes, it does bring, you know, the sleepless nights and the colds and all of those things, but you put everything aside because the richness, those little moments are priceless.
They are, absolutely, without a doubt. Yeah.
So tell us about your book. I did not know you wrote a book. How did that happen?
So I wrote a book. So this is a big shout out to Terry Kanakin. So when I was at Wyeth, and I was working on the FSH receptor and stumbling on signaling bias, I had the opportunity to attend a Nature Summer Symposium in Miami on cytokine receptors. And Tony Possum was given a talk there, and he's talking about how all these different proteins interact with cytokine receptors to induce a physiological response, right? And I had hypothesized when I was working on FSH receptor that really GPCRs are really bimodal in nature. You know, so you have a ligand binding pocket, and then but it has an inherent affinity for a ligand, and then you have on the cytoplasmic surface a binding pocket for the signaling effector that has its own inherent affinity, and depending upon the conformation stabilized by the ligand, that alters the affinity of the binding pocket on the cytoplasmic surface. We now know that that's pretty much how it happens. But at that time we didn't know anything, and that was my hypothesis. And so I went to see this talk with Tony Possum, and he was talking about this stuff, and he was talking about the differential affinities for cytokine receptors to these different associated cytoplasmic proteins like GRAB and SOS and stuff. And I said, it just, you know, light bulb, it was a pure light bulb moment. He said, I thought, all these receptors are evolutionarily conserved. They all function the same way. So, but I had no data to support that, and I had no time to actually explore that because I was, you know, I had programs to progress.
Yeah.
So I sort of put it in the back burner, but it had always been in the back of my mind that, you know, over time, hopefully people would come out and come to that realization. So long story short, in 2011, I started to see this data start to come out in the literature. And I don't think people really understood it from across the different areas. So Terry Kanakin was visiting BMS to give a talk on GPCR pharmacology, and I knew Terry, and I walked up to him before the talk, and I said, "Hey, Terry, I was wondering if you'd help me write this book." And Terry, classic Terry Kanakin, he goes, "Yeah, sure, great." And then we started discussing, and I says, "So Brian, what's your book about?" It's just, he's such a super nice guy.
He is.
And and I said, well, I have this concept that all receptors function pretty much the same, and it's evolutionarily conserved from the get-go. And I want to write a book because nobody's teaching it this way, right? And I think the data's out there. And he said, "Oh yeah, absolutely. So let's do it." So he hooked me up with Elsevier, and so I proposed my book concept to them. They loved it, and so we wrote this book. It's called Bias Signaling in Physiology, Pharmacology, and Therapeutics. But the real premise of the book is about how receptor function is conserved across receptor classes.
Wow.
And how signaling bias can be found in every receptor class. And now we know that that's absolutely true. And the reason I wrote a book as opposed to writing a review is I kind of wanted to get around peer review. It sounds terrible, but I really, I knew, because you have to understand your place in the greater scheme. And here I was, some guy that really wasn't that well known at the time, but I had this cool concept. So I wanted to, rather than writing a review and leaving my fate up to others, I figured I'll just take the bull by the horns and write a book instead. And so I have. It's actually sold pretty well. I think, it's done okay. I published it in 2014. And I would encourage anyone who listens to this to read it because it's really cool.
I will definitely put a link to your book on the podcast page when the podcast airs. Yeah. Which will most likely be early next year.
Thank you.
But definitely, I think it's a great way to put your thoughts on science and on a topic that passions you into something palpable that people can go back to. And Terry Kanakin is just phenomenal. I've met him several times. Last time, I think it was 2019, he was giving a course on signaling bias and GPCR function at the Discovery on Target here in Boston.
Yep.
And I have one of his books, so I asked him to autograph it, and he was extremely humble about it. And I don't think he felt very comfortable, and I didn't want to push it and ask for a picture with him. I said, next time I'll get a picture with you. But thanks for
humility is really, he's a really humble guy. And that's another thing I would give a little piece of advice if you want it for young scientists is remain humble because, humility precedes promotion, as they say. So I really feel that that's an important aspect. And as long as you remain humble, things will come your way. Karma exists. And
yes, I agree.
And I really feel like Terry is just, he's I would call him a friend now. We don't talk as often as maybe we should, but I really, really, he's another guy who's influenced my career, whether he knew it or not. and I'm really, really grateful for it.
I think he influenced a lot of careers with all his papers. I remember when I started reading his papers, and it took me a week just to try and understand what it was all about. And you could see that. I think I saw him. Oh yes, I saw him at a conference, and on paper, while he was listening to a talk, he was doing some magic math. I told myself, oh goodness. And it seems so easy coming from he's
a treasure. I mean, he really is. He's I would encourage you to, because he is so approachable, and I've really benefited from approachable scientists my entire career. Terry's one of them. I would say Michel Bouvet is another one. I can't say enough great things about Michel because he's had a great influence on me too, whether he knows it or not. And he's always has time to talk to you. It doesn't matter who you are. Yeah.
That is true.
He'll sit down and chat with you.
That is true. So Michel was one of my professors in undergrad at the University of Montreal back in the day. And we're still in very loose contact once a year when I wish him happy birthday or happy new year. And I'm so excited because he's extremely busy, so I've been trying to get him on the podcast since July, and now we're in November. And next week, actually on Wednesday, I'm recording an episode with Michelle, which is
fantastic.
Wow,
I'm really honored. Holy
I'm really excited about it, and I'm also excited. So it's a secret, but I'll try to tweak it. But I will be recording a podcast episode with Bob Lefkowitz as well.
Fantastic.
Which
That's awesome.
Made my year, I have to say.
Of course. I mean, really, he's you know, and I don't know him very well. I've met him on a few occasions, and I would say we're acquaintances. But obviously, everybody who's a GPCR file has followed his work, right? I mean, he really is he's the quintessential GPCR pharmacologist of our day.
Yes.
And if anybody deserved the Nobel Prize, he did, no doubt.
Absolutely.
He's really impressive. He's inspiring.
He is.
You know, I actually, I think I'll have to tune in to see his life story, because I want to know his perspective.
Yes, I think that the whole podcast is going to be just phenomenal, because everyone keeps telling me, you know, he has great stories, and I know I've seen him give talks, and I've met him a couple of times here and there. I even have a picture with him. He may not remember me, but I have the picture from, I think it was 2013. So he was supposed to come and give a talk in 2012, the week of the Nobel Prize announcements. And yes, I was a postdoc at Rockefeller back then. And obviously, well, he didn't come because the day before he learned that he got the Nobel Prize. So we waited a year to reschedule that lecture, but I think it was just absolutely worth waiting. And I'm trying, and I'm going to put this in here, but I'm going to ask you offline as well. I absolutely want to have Terry Kinakin on the podcast.
You should.
And
without a doubt.
I've tried, so I may need a little push to get him. I know he's very humble, but I think the GPCR field would absolutely benefit from hearing what he has to say.
Absolutely. And you know, another person I really had an influence who was very approachable was Martin Rodbell. So I had the opportunity to meet Martin through Andres, who had worked with him at the NIH. And so it's really, the story is that so Martin was giving a talk at Wyeth when I was there, because Andres had invited him. And we went out to dinner. So it was Paco Lopez, Francisco Lopez, myself, basically Paco's lab, and Martin. And so Paco puts me on the spot and says, "Brian, show Dr. Rodbell your data." I'm like, "You gotta be kidding me, right?" So anyway, I draw it out on a napkin, you know, during dinner, what we saw with, he goes, and his response was classic. He goes, "I'm not surprised." That's what he said.
Oh, God.
He said, because he goes, "I actually proposed this in one of my papers in 1972. And I'm like, well, but he, you know, he was very, very encouraging, and he goes, you absolutely have to follow up on this research. I have no doubt that this actually happens. And so, you know, he again was another guy who's very, very, you know, I was 32 at the time, 33. And, you know, I was just starting my career, and here's this Nobel Prize winner. He had just won the Nobel Prize in '94. And you know, I was just in awe. I mean, yeah, I'd never met a Nobel Prize winner before. and so I just thought it was the coolest thing. And it was, but you know, these people, they really have a power to inspire younger people when they approach it correctly. And when they realize that, you know, that people look up to them, and they really are looking for inspiration from them. And if they embrace it, man, it's powerful.
Yeah. And I think that's one thing I realized starting the podcast, and also I think getting also a little bit more experience, not to say that I'm getting older, is that people are so approachable, and so there are people that are more approachable than others. But don't be afraid to go out there and, you know, ask for a 5-minute phone call or ask for an email exchange, as long as you can add value to that person. And you'll be surprised how nice people are.
I love it when the young people call me and you know, write me an email. "Hey, can we chat about career opportunities?" I soak it up, because it really is a chance for me. Not being in academia, I don't get many opportunities to give back. Mm-hmm. In that way, you know, so at least in a classical sense. And so when they do, I get really, really excited. I jump at those chances because it's really rewarding, you know.
Well, there you go. You have an open invitation, the audience, an open invitation to find you on LinkedIn.
Actually, send me an email, brian.arey@bms.com, and I'll be more than happy to find time. I'm probably opening myself up to a flood.
Maybe, maybe, who knows? So here's my question. When we're talking about giving advice to young scientists, what would be your top 3 to top 5 things that you'd like them to know at this point?
Well, okay. So I think they're not going to be in any particular order because it's hard. I haven't really thought about priority.
It doesn't matter. I think as long as they're on point and they're actionable, we'll be happy to take them.
I think one of them I've already alluded to, and that is always keep an open mind, and trust your data. Ask tough questions of your own data. That's another one, right? Don't be afraid to challenge yourself scientifically, because through that's when you really come across valid discoveries. The other one would be give back to others. I think it's really important to see yourself as a greater citizen within a global community, and always give back to others as best you can. I follow the data. That's another one. Just really don't. Let your data choose the next experiment. That's the way I've always approached it. Another one is again, and I alluded to this at the very beginning of our discussion. It's really about technologies, and we have a ton of them these days, and they're just exploding. Technologies are tools, and apply whatever technology you want. So always try to question what you can do with a certain technology, and that's one of the things I really incorporate within my team. Is that let's really see if we can be disruptive. I'm not a huge fan of antibodies.
That's a tough thing to say in this day.
I know, but antibodies are from a technological standpoint,
yeah.
They've been very useful tools, but let's face it, that's 50-year-old technology, and we can do better in terms of assay development. And so that's one of the things I sort of harp on my guys. Let's think of new ways of doing it rather than be, beholden to antibodies all the time. And then the other one, I guess, would be really to persevere, and don't be afraid to persevere. We all go through challenges. Your career path is not going to be easy. And things are going to happen. You're going to run across people who aren't really nice. It just happens, and you just have to persevere through it, and have confidence in yourself, as you said very, very early on. So those are the things it's really, I guess there's no really true insights about science in itself other than I think, because I think science is really a personal endeavor. It's a creative, personal, and I hate to say it because this is so overused, but it really is an art form, and everybody approaches it differently.
It is. It's funny because I think you're the first person who says this openly, that it's an art form. I always thought that it was an art whenever I was doing an experiment in the lab, and everything worked perfectly. The cells, and I'm really downsizing it to the little minute details. But when you come in the morning and your cells are glowing and they're looking beautiful, and then you run through the entire assay, and then you analyze the data and you get your sigmoidal dose response curve, and you look at the triplicates and you're like, even if I wanted to come up with numbers so close to each other, I couldn't have. That's an art. That's beautiful.
Without a doubt, because not everybody can do that. Trust me.
but
it seems very simple, but it's not easy. Right?
I think it's a matter of practice, and it's also a matter of marrying the right question to the right assay, as you mentioned before.
Yeah. So one point I would like to make here, and I'm sorry if I'm sort of diverging a little bit.
Not at all.
I think we all have to understand that what we study in terms of pharmacology, whether it's a GPCR or whatever, what we're really studying is evolution, and at the base level, and when you think about it from that perspective, it's hard not to be awed by nature. Just the simplicity of a sigmoidal dose response curve, and thinking about how that happens and how that, how Mother Nature selected for that. It gives me chills sometimes. It sounds silly, but really I look at it, and when you see, like you said, when you see this perfect sigmoidal dose response curve, it's like, wow, that's amazing.
Exactly. And then you take it to the next level and you have people like Terry Kinakin who can actually associate equations and math to it.
Right.
And then you're like, how is this even, how does Mother Nature do this?
It's really, really incredible.
I think so too. And it's funny because we were talking about, evolution and people being genetically different on different aspects, and about finding drugs that kind of more fine tune different aspects of a pathophysiology. One question I had is, so we have the hardcore drugs that you would take that would most likely, billions and billions of dollars, years and years go into research, but what are your thoughts about more natural methods? I know that we don't have enough data on natural compounds or, food-based, diabetes management and things like that. What are your thoughts on that?
So my thoughts on that are pharmacology is based on those things. If you look at history, and I actually, in the first chapter of my book, I talk about this, right? I'm not the first one to make this connection, a lot of people have over the years. I'm just another one. But the matter is that pharmacology really had its roots, and the treatment of disease had its roots in oral history and therapies, plant-based therapies that were handed down from generation to generation. And so these go back thousands and thousands of years. I don't refute them. I think that what I do get a little frustrated with them is that a lot of times I think they are presented in a way that is not necessarily scientifically rigorous. Right. So there's many aspects to drug discovery that people aren't aware of. And so when I see somebody saying, you take this protein, it's going to have an impact on your joints, and you take it as an oral drug, I mean, come on.
Yeah.
That doesn't make any sense to me, because the fact of the matter is that, this is it's going to get digested, et cetera.
Exactly. But then again, I think, please go ahead.
No, go ahead.
I was just going to say that sometimes I think the one of the issues with all these, for example, natural supplements and things like that, is because they're not regulated. There are not rigorous studies linked to it. And since it's not regulated, one brand versus another is very different. So you have differences in additives. And what influences the most is, yes, the person's genetics, but also what they eat and how they live. And I think those things are extremely difficult to control.
Yeah, and I think, natural products has never completely gone away from the drug discovery realm. It's still there even today. So we still look for natural products because there's a reason for that, because they exist. Taxol's, I guess the poster child for that. And I think that they exist, and we should try to explore them. But I think at the same time, we need to do the science rigorously and using all the things that we understand about how drugs work within the human body. And so I think, yeah, I do not poo-poo them at all. I think we certainly should do that. I just think, and again, it all comes down to dose response rate. Just because something has a micromolar effect on a isolated cell doesn't mean it has an effect in the human body.
Definitely agreed on that. So before I let you go, so you mentioned people can reach out to you. I will share your email, share your LinkedIn, and we'll definitely take up on that. If you had an advice to give, I know we talked about advice to young scientists, but from a scientific perspective and from a career perspective, but let's say someone is in academia and they're looking to move over to industry, what should they think about first?
Well, I think if I was them, I would really try to understand in detail drug discovery and basic pharmacology. But I mean, you'd be surprised. There is actually a dearth of trained pharmacologists within early research. And I don't know why that. I think, well, I do know why it is. I have a personal opinion about it, but I think there was a movement to get away into more subject matter experts within therapeutic areas over time, and the classical study of pharmacology has sort of been put off on the back burner. I'm not a classically trained pharmacologist, but I certainly have appreciation for it. And so I try to, when I look for people to hire, I always look for somebody that has a pharmacological background, because we need that. We really need that expertise. So I would, don't be afraid to apply for a job. You never know.
Yes, because sometimes I feel like the job descriptions are not as accurate as you'd like them to be. And also sometimes, based on previous experience, sometimes hiring managers not necessarily, not all the time, but sometimes they don't know what they're looking for until they see it.
No, that's really true. The other aspect of that, and I'm sorry if I'm long-winded.
No, of course not.
No, is that, the fact of the matter is, what you need is somebody who's a good scientist. Most people can learn things, and if they have a desire to learn, you can plug them in almost anywhere. I hate to say that, but it's really true. People can apply their expertise to almost anything, and be successful at it. So whereas job descriptions are generally written in a very specific fashion because they have an idea of what they want. I've hired people that didn't fit the job description before because I thought that they were the best scientist, available. And, right now, the job market for scientists in the drug discovery space is very competitive. So,
Yep.
It's definitely, a candidate's job market right now, not the employer.
It is. And I think based on how the market looks like now, and when you were thinking of going to industry, right, it's so much more active now than back in the day.
Night and day, and it was that way my entire career, really, up until the last 3 or 4 years.
Wow.
Really, the influence of venture capital into the biotech space has really made it an attractive job market.
It is, and I think the sky's the limit whenever you're following the science. So,
yeah.
And also find the right team, find the right people. And I love the fact that you mentioned that you're hiring for potential, which is amazing.
Absolutely. Yeah, you have to.
Well, with this, Brian, I want to thank you for your time. I loved our discussion. And I'll definitely be in contact, because I really want to have Terry Kinakin on the podcast.
Oh yeah. So if maybe I can grease the wheel a little bit.
Thank you. I'll use my quote unquote GPCR podcast celebrity here.
I don't know about that, but okay.
No, I think my cachet with you today and with our previous guests to get even more guests on the podcast. Thank you so much, and then take care, and we'll talk to you soon.
All right. Thank you.
Thank you.
I need a favor. Please share the Dr. GPCR University program with your friends and colleagues, especially if they live and work in a developing country. Please subscribe to our YouTube channel. Many of you come back to watch our videos but aren't subscribed. Having more subscribers will help us get you more content. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest and our Dr. GPCR team, Attila, Ines, Montserrat, Ivana, Cam, John, Yatsu, Andreina, and Balint. If you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. And don't forget to subscribe to our monthly newsletter on our website to get all the news about all the work that we've been doing and we are doing at Dr. GPCR directly delivered into your inbox. Another great way to support us is to share your favorite Dr. GPCR programs with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com or hello@drgpcr.org. As I mentioned, both emails will be active for a foreseeable future to make sure that we don't miss your email. Until next time, stay safe.